Okay, chapter 5, part 2. I'm gonna cut a little bit of this stuff out for you guys. Okay, so I am being recorded. Show from the current slide. Okay, so I'm gonna skip all this like generation of ATP stuff. Okay, I'm gonna move to metabolic pathways of energy production. Okay, so okay. Carbohydrate catabolism. Okay, I'm going to break down carbohydrates. That is the primary source of energy, especially for prokaryotes, especially for bacteria. Carbohydrates are the major source of energy. Okay, all right. Okay, carbohydrate catabolism. Okay, the breakdown of carbohydrates to release energy occurs in 3 principal stages: glycolysis, the Krebs cycle or the citric acid cycle, and the electron transport chain. Okay, what you get out of this is not every single step in the process, nope, I'm not going to ask you for every single enzyme or product in the process. What I do want you guys to understand is a big overview of the 3 steps. What goes into each of the 3 steps and what comes out of each of the 3 steps. Okay? Okay. So the first thing that happens is glycolysis, breaking down of glucose, okay, into pyruvate or pyruvic acid. Okay, pyruvate and pyruvic acid mean the same thing. Okay, this step produces ATP, which is energy, and NADH, which is a reduced molecule that's capable of producing energy down the line. Okay, so okay, so this is glycolysis. Oops. Okay, share screen. Oh. Share sound. All right.
Metabolism of glucose is of central importance to all living organisms and is in fact performed in almost every cell. As one of the major sources of energy in living systems, glucose is made and broken down constantly in living cells. The metabolic pathway for the breakdown of glucose is called glycolysis, from the Greek glucos for sweet or sugar and lysine for breaking apart. Glycolysis is sometimes called the Embden-Meyerhof pathway after its discoverers. Glycolysis consists of 2 main phases, the preparatory stage and the energy conservation stage. The preparatory stage uses 2 molecules of ATP to phosphorylate glucose, making it easier to break it apart into 2 3-carbon molecules. In the energy conservation stage, 4 ATP and 2 NADH molecules are produced, along with 2 pyruvic acid molecules. At the end of glycolysis, the energy found in glucose has been converted to a net of 2 ATP molecules and 2 NADH molecules.
All right. So, okay. Don't worry about the preparatory stage and the Embden-Meyerhof pathway. Okay. But you should know what is the end product. Okay.
During the preparatory stage of glycolysis, energy is invested in the form of ATP to prepare glucose for utilization by the cell. This stage can be further divided into the energy investment stage, during which the ATP is used to phosphorylate glucose, and the lysis stage, during which the doubly phosphorylated glucose molecule is cleaved into two phosphorylated 3-carbon sugars. Let's look at the energy investment stage first. While glycolysis results in a net gain of available energy for the cell in the form of ATP and NADH, energy must first be invested to begin the process. This can only occur in a living cell with ATP available to invest. In the first step of glycolysis, a phosphate group is In the second step of glycolysis, the atoms of glucose 6-phosphate are rearranged to form fructose 6-phosphate. This rearrangement prepares the molecule for the third step of glycolysis, in which further energy is invested in the form of ATP, forming fructose 1,6-bisphosphate. In the fourth step of glycolysis, fructose 1,6-bisphosphate is cleaved into two 3-carbon sugars, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This step is the lysis step for which glycolysis is named. In a very rapid reaction, dihydroxyacetone phosphate is converted to glyceraldehyde.
You don't need to know this. All right, so you do have to know this. The net gain is 2 molecules of ATP plus 2 molecules of NADH. So this equation is what I'm asking you guys to know. What goes in and what comes out, right? So here's the arrow. Okay. So don't worry about the other pathways, alternative pathways, we're skipping that. Okay. Okay. The next step is cellular respiration. Okay. Now I can go one of 2 ways. Okay. So I've broken it down in pyruvate. Now I can go either the aerobic pathway if I have oxygen, or the anaerobic pathway if we don't. Okay. In humans, we prefer the aerobic pathway, and there's reasons why. But in bacteria and stuff, certain types of bacteria actually prefer the anaerobic pathway, and there's reasons for that too. Okay, so we'll get through with that later. Okay, so here's the Krebs cycle. I'm gonna skip the animation because I don't think that's really that important. What you do need to know out of the Krebs cycle is that you have CO2 produced. That's why you're breathing out. You have a whole bunch of NADH produced. Okay, that's going to be high energy molecules that I will use later, and FADH2, again, same thing, high energy reduced molecule that I will extract energy from later. Okay, the next step is the electron transport chain. Okay, the electron transport chain. Okay, I'm going to transport, okay, electrons, okay, now the electron transport chain, and along that time, okay, ultimately, I'm going to put the electrons onto oxygen, okay, I'm going to drop those electrons onto oxygen. So without oxygen, I don't have anything to drop it onto. So without oxygen, I'm not able to use aerobic respiration. Okay. Okay. When I passing down an electron transport chain, I actually actively pump hydrogens in between the 2 mitochondrial membranes. Mitochondria have an inner membrane that's really kind of folded, and an outer membrane that's just kind of a big circle. I trap electrons between those two membranes. Those 2 membranes are fatty acids, so it's going to be hard for electrons to run past the membrane, so they're stuck there. The only thing that gets them through is this little thing called ATP thin phase. Okay, ATP thin phase will allow electrons to pass through the membrane, but it uses the energy of allowing them to pass through to make an ATP. Okay, so as I come through, okay, for every NADH here, and I make 10 of them. I just, this is physiology. This is not physiology, right? So you make a bunch of NADHs, and NADHs are trapped here, and they get turned into ATP here. Okay. Okay. Make sure you understand the final electron receptor in the electron transport chain is molecular oxygen. This is why we breathe, and this will play a role later. Okay. The overall aerobic respiration figure also need to know this. Okay. The whole reason why we breathe oxygen, okay, is I take glucose, I take oxygen, and I end up with 38 ATP, and I break it down in the carbon dioxide into, or I break down the glucose into carbon dioxide and water. Okay. All right. Now I'm going to explain this in terms of oxidation and reduction. Reduction is having hydrogens, right? I explained, like, reduction is having electrons and hydrogens. Okay, I have a whole bunch of hydrogens here, the H12. I lost all my hydrogens here, so the glucose got oxidized. Okay, if something gets oxidized, something else just needs to be reduced. So we'll put that one in blue. Okay, oxygen gets reduced to water. That is reduction. Okay, the sugar gets oxidized in carbon dioxide. The oxygen gets reduced to water. This oxidation reduction reaction generates 38 ATP. Okay. Okay. Anaerobic reactions. Anaerobic reactions, the final electron transport chain is acceptor is not oxygen, and therefore it yields less energy than aerobic respiration. Okay, so here are some of the electron receptors. Okay, so they end up with different amounts of ATP, but not the maximum 38, which is what oxygen are at. Okay. Okay, so make sure you understand this part right here. Okay. Okay. Make sure you understand these things. Okay. Products. You don't need to know 5.14, but these two you do. Okay. Anaerobic respiration, you get much less. Okay. In fact, one of the types of anaerobic respiration is called fermentation. Fermentation does not even require oxygen. Okay, so you can ferment things without oxygen. In fact, if you make beer, okay, like the most famous fermentation is beer or wine, and you make beer and wine by putting in grape or wheat sugars and put it in yeast and seal it and let it grow in a little container sealed. and that sealed container doesn't have oxygen, so you get fermentation. Okay, fermentation. You go from pyruvate and you add the NADH. Remember, we have NADH, but NADH can't be put into the electron transport chain because there's no electron transport chain without oxygen. So instead, you just dump the hydrogens and electrons back onto pyruvate, and you end up with lactic acid or other components. Okay. The other component that some things produce are alcohol, and this is what we use it for. Okay. So fermentation, we break it down into lactic acid. Other organisms, they're able to remove one of the CO2s and break it down into ethanol. Okay. And other organisms break it down into other stuff, and we extract this stuff. Okay. Yeast, we breaks it down to ethanol and CO2, and that's why beer is bubbly and stuff like that. Okay. Other bacteria break it down into lactic acid, which we use, acetic acid, which is vinegar, okay, and other stuff. Okay. Okay. And this is what we get out of it. Okay. Okay. Lipids and proteins, I'm gonna cover really superficially. Okay. Lipids break down into something called fatty acids. Fatty acids go into something called acetyl-CoA, which is in the middle of the Krebs cycle. Okay. Proteins, depending on which amino acid you have, they enter into different stages of the of this process. Okay. Okay. All right. So I can use biochemical tests to detect which enzymes they have, and which enzymes they have will tell you what type of bacteria it is. Okay. So we'll do this lab virtually. We'll detect amino acid catabolism enzymes in the lab. If they're able to catalyze the reaction, they turn the reaction purple. Okay, they change, the thing becomes more acidic and turns purple. If they don't, then they stay this color. Okay. The other test that you will do in the lab is the oxidase test. Okay, oxidase test identifies bacteria that have cytochrome C oxidase. Okay, and they're able to break down one electron. They have an enzyme called cytochrome C oxidase, and that shows that they have an electron carrier and they're capable of using electron transport chain. Okay, like an example, Pseudomonas. Okay, so here you can see the fermentation. Fermentation makes things acidic. So you can see these. This is a control. This is acidic, and you can also see there's a bubble here. That bubble that's not here. You see a bubble here that shows it's making carbon dioxide. Okay, we'll do the fermentation as well. Okay. All right. Pause for a second. Okay, don't worry about this biochemical test. We'll go over it more in the lab, so that don't worry about it for lecture material. Okay. Photosynthesis. Okay. Learning objective 520 is the only one I want you guys to know. Okay. So photosynthesis breaks down into two things: light dependent reactions and light independent reactions, otherwise known as the light reactions and the dark reactions. Okay, the other term for the dark reactions is called the Calvin cycle or Calvin-Benson cycle. Okay, so photosynthesis, photo, light, synthesis, to make stuff. I'm using light to make stuff. So I'm converting light energy into chemical energy. Okay, so the light reactions, I'm using light to make ATP and NADPH. NADPH is highly reduced, so highly energetic. So I can use those two high energy molecules to turn carbon dioxide into sugar. I am reducing carbon dioxide into sugar. Just like when I burned sugar, I oxidized it to carbon dioxide. Now I'm reducing carbon dioxide back to sugar. Okay. So don't worry about the oxygenic versus anoxygenic. Okay. Okay, so you can watch this video a bit. Oh, sure.
Photosystem 1 is composed of a large number of chlorophyll molecules, which harvest light and pass the excited electrons between the chlorophyll molecules until the excited electrons reach the reaction center. After the excited electrons are passed to the reaction center, these electrons are passed to the next protein in the electron transport chain. In the cyclic photophosphorylation pathway, the electrons that are released by the excitation of photosystem 1 pass through 3 sets of electron transport proteins, finally returning to photosystem 1. On their way, the electrons help establish a proton gradient as they pass through a group of proteins called cytochromes. The proton gradient in turn drives ATP synthase to produce ATP through chemiosmosis.
This is the Calvin cycle. This is the light reaction.
The Calvin-Benson cycle, sometimes called the light-independent reactions or dark reactions of photosynthesis, is the enzymatic pathway that uses the chemical energy of ATP and the reducing power of NADPH to generate glucose and other sugars from water and carbon dioxide. Through 2 rounds of the Calvin-Benson cycle, a single glucose molecule is produced from 6 carbon dioxide molecules and 6 water molecules, at the expense of 18 ATP molecules and 12 NADPH molecules. In the first step of the Calvin-Benson cycle, 3 carbon dioxide molecules from the air are covalently attached each to one 5-carbon ribulose bisphosphate molecule. The 3 resulting 6-carbon sugars react with water and break apart into 2 3-carbon sugars each, resulting in 6 3-phosphoglyceric acid molecules. This step is called the carbon fixation step. In the second step of the Calvin-Benson cycle, 6 molecules of ATP are used to phosphorylate each of the 3 phosphoglyceric acid molecules produced in the first step, resulting in 6 1,3-bisphosphoglyceric acid molecules. 6 NADPH molecules are then used to reduce the 1,3-bisphosphoglyceric acid molecules to 6 molecules of glyceraldehyde 3-phosphate, G3P. The ATP and NAD.
Right. So basically, the Calvin cycle or Calvin-Benson cycle, carbon dioxide is turned into sugar. You take the energy from the light reactions, the ATP and the NADPH, okay, the ATP right here and the NADPH right there. Okay, the ATP and the NADPH, and you convert that into, you convert that into sugar. Okay. All right. Oops. Hello. Okay. Okay. All right. So, so make sure you kind of understand this. Okay. So here are the energy sources. So light, glucose. You use the electrons to generate ATP, and then you put them on electron carriers, which then you generate more ATP. The final electron acceptors are going to be the ones accepting the electrons. Oxygen, okay, organic compounds, fermentation, etc. Okay. All right. Metabolic diversity, we're going to go over. So make sure you understand what these terms mean. Okay. So I need two things. Okay. I need energy and I need material. If I want to build something, I need the energy to build it and I need the material to build it with. Okay. So I need the energy source. The energy source can either come from chemicals or from light. Okay. If it comes from chemicals, we're chemotrophs. Okay. If it comes from light, phototroph. Okay. Now, the second thing we need is the material, the carbon source. We build things out of carbon. Remember, study of carbon, organic chemistry is a study of carbon. Okay. If the carbon comes from an organic compound, any organic compound, then it is heterotroph. Okay, hetero is different, so different things. Okay, different organic compounds, heterotroph. Okay, if it comes from CO2 and we make it ourselves, we make the organic compound from CO2, then autotroph, self-making organic compounds, right? We make it from something that's technically inorganic, CO2, so autotroph. Okay, so we can be chemoheterotrophs. Okay, we can be chemoautotrophs. Okay, we can be photoheterotrophs, and we can be photoautotrophs. Okay, you don't have to know anything below that green line. Okay. Okay. Yeah. So, okay. So basically, you have to know this chart, right? So that graph is explaining what this chart is. Okay. Okay. We'll skip all this, the biosynthesis parts. Okay. Forward. Okay. So that's what end this chapter. I tried to shorten it because it was way too long. Okay. All right. This is the end of chapter 5. So the material on this chapter, everything from here to the very beginning of the course will be on your first exam. Right. So good luck. Happy studying. And email me if you have questions. Okay. I'll probably send a review for you guys. So, but we'll talk about that later.